Okay, MERS, Middle East Respiratory Syndrome. I want to first talk a little bit about emerging viruses because that's what this is, right? What's an emerging virus? Now, this is a little bit from the course, but it doesn't hurt to repeat things, right? An emerging virus is the causative agent of a new or previously unrecognized infection. So two possible definitions. This is a term coined in the 90s, but of course emerging viruses aren't new. We just have been able to discover them. Since the rise of agriculture, 11,000 years ago or so, new infectious agents have invaded human populations because they can be sustained by numbers that were unknown before agriculture and commerce. We lived in hunter-gatherer packs, few hundred people, and that doesn't sustain infectious agents. But as soon as we've learned how to do agriculture and more and more people could live together, getting food, then population numbers grew and infectious agents begun. And as I said, we've recently gotten really good at detecting emerging viruses. A little more definition. An emerging virus can have an expanded host range with an increase in disease not previously obvious. For example, poliomyelitis for much of human history was a rare disease, never caused epidemics. And then around 1900, boom, epidemics. So it's a increase in disease, and understanding why is, of course, important. Now, sometimes, or often, every time there's a new virus, it comes from an animal. And a virus goes from a wild or domesticated animal to a human. It's called a zoonosis. The zoo, the animal part, that's a zoonosis. And sometimes these spillovers from animals to humans establish a new virus in the population, a new human virus. For example, SIV moving from chimps to humans, that's now HIV-1, a human virus. Or SARS-CoV-2 from bats to humans, that's now a new human virus. But not all crossovers, not all emerging viruses establish themselves as human viruses. Often, cross-species infection cannot be sustained. For example, every time there's an Ebola virus outbreak, it's a new spillover from bats or some other animal reservoir to humans. MERS coronavirus, as we'll see today, typically new spillover from camels to humans with limited human-to-human transmission. Now remember, Ebola virus in West African outbreak 2015, over 20,000 cases, obviously a lot of transmission, but it was a special case because the transmission mainly occurred in hospitals and at homes. So there was no community transmission, people walking around spreading it in theaters and sporting events and restaurants like SARS-CoV-2. It doesn't happen with Ebola virus, doesn't happen with MERS-CoV-2. Now, I love this graph. Ancestral origins of human pathogens. So these are four bars on this bar graph. Now, the y-axis is the number of viruses belonging to genera. So these numbers in here are genera of viruses, not species, but a little higher order, genus. And then on the bottom are the different categories. So we have 31 viruses, members of 31 genera that are human viruses, but they came from animals. So poliovirus and herpes simplex virus, all of these human viruses, HIV-1, they came from animals at some point. Members of 37 genera are zoonotic pathogens. They continue to spill over from animal reservoirs into people. Ebola virus, Nipah virus, even West Nile virus. It doesn't become a human virus. It doesn't transmit from human to human. It goes from mosquito to human, and the mosquito gets it from a bird or a rodent reservoir. Now, the next one, 16 members of 16 viral genera. We got from ancestors of Homo sapiens, right? The hominid ancestors. They got the viruses from animals, and then they passed it on to the Homo species. And then finally, members of 6 genera, we acquired from our Homo relatives, Homo sapiens from Neanderthalensis and Denisovensis. Is that the right way to say it? Denisovans. They got it from animals and they passed it on to us. Every virus came from nature. And if you want to ignore history, well, then it's your demise because the facts are all there. Okay. Now, why is this that viruses emerge? And let me move this little thingy here. Overriding factors driving the emergence of infectious diseases. Well, number one is population. Look at the population growth. Billions on the y-axis and years on the x. Look at this incredible growth. Population growth everywhere, invading all ecosystems. And this makes us contact animals which have their viruses, right? That's just the way it is. What else? Well, anthropogenic activities. Humans continuously devise new ways to encounter viruses. Dams, water impoundments, irrigation, deforestation, transport of animals, blood transfusion, air travel, urbanization, daycare centers, hot tubs, air conditioning, use tires that carry mosquitoes, climate change, natural disasters. Look at that. Look at this snapshot of all the flights in the air at any one time. Amazing. So we have devised ways to encounter new viruses. Don't blame nature. Blame humans because we set it up. Okay. Finally, it's important to understand that evolution plays a big role in the emergence of pathogens. Because as viruses multiply in their hosts in nature, whether it's a bat or a pig or a duck or whatever, they mutate. And so virus populations are quasi-species. They're composed of millions and millions of mutants in any one animal, many, many different mutants, one of which may randomly be able to infect a human. And should that virus encounter a human, then it could start a pandemic. But many viruses exist that could infect humans, but they never encounter humans. Now, SARS-CoV-2 existed somewhere in a bat, and it was brought into an animal market and contacted humans. So we facilitated its contact with humans by bringing in animals from the surrounding area to Wuhan that had been in contact with bats, which had the virus. And then in the meat market, it spread to humans. So you have lots of mutants existing in the animal reservoirs, and those mutants allow adaptation to new hosts and environments. That's how evolution plays a role in emergence. Okay, that brings us to MERS and MERS-CoV. MERS. So there's two names for this. Just like HIV/AIDS, just like COVID, SARS-CoV-2, MERS, Middle East Respiratory Syndrome, and MERS coronavirus is the virus. The disease is MERS. MERS-CoV is the virus. You know, when COVID and SARS-CoV-2 were first named, people were like, oh, why do we have to have two? Well, just look at what's been happening in history, for God's sakes. We do that all the time. Don't complain. So the first case ever seen of MERS, 2012 in KSA, a 60-year-old male patient died of pneumonia and renal failure. They suspected virus infection, recovered virus from this patient. They sequenced the genome immediately. They identified it as a new coronavirus, a novel coronavirus. They identified the receptor for this virus, dipeptidyl peptidase 4, which interestingly is not present in the human upper respiratory tract. Okay? So that's going to tell you right away why this virus does not transmit well among people. If a virus is to transmit among people efficiently, it has to reproduce in the upper tract because that's where you're shedding virus all the time. You don't shed a lot of virus from the lower tract. You know, when they tell you to give me a really deep cough, we want mucus from way down in your lung. It's hard to do, right? So without upper tract reproduction, it's hard for a virus to spread extensively. Now, it doesn't say that it won't change at some point. It could change receptors, perhaps, but it hasn't. So this virus was rapidly identified, sequenced, and found to be closely related to bat coronaviruses, although there is a phylogenetic gap. This we'll see in a moment what I mean. And it's not SARS-CoV. It's distant from SARS-CoV. It's a different virus because, of course, as soon as they saw it was a coronavirus, people started to worry, but it's not SARS-CoV. So in terms of the origin, two kinds of bats carry coronaviruses similar to MERS coronavirus. The polymerase, RNA-dependent RNA polymerase of MERS-CoV has 92% amino acid identity to the pipistrelle bat HKU5, and the S and the N genes are more closely related to a different bat virus, HKU4. But it doesn't prove that these are the ancestors of SARS-CoV-2. It just says they share a common ancestor. Now, more recently, an interesting virus was discovered in South African Neuromycia capensis bats that has 85% similarity with MERS-CoV, very, very close. And it's called NeoCoV. Doesn't cause MERS, but it's close, and we'll talk about this in a moment. Well, right now, this paper published in 2022, which I think we did on TWIV, this virus, NeoCoV, close relative of MERS coronavirus, right? NeoCoV, South Africa, it binds ACE2, not DPP4. So what is that telling you? This ACE2 we know is in the upper tract. SARS-CoV-2 binds ACE2. It spreads really well because it reproduces in the upper tract. So it's possible that NeoCoV could eventually emerge as a pandemic MERS virus, but that hasn't happened yet. But other things have to be in place as well. But the first step, having the receptor in the upper tract, is pretty important. Okay, so here's a phylogenetic tree of some coronaviruses to put this in perspective. So again, this means how they're related on their genomes. So down here is MERS coronavirus, and it's close to some bat coronaviruses that I just told you about, HKU4 and 5. And then a good distance away is SARS-CoV, and a good distance away is SARS-CoV-2. About 60% identity with SARS-CoV-2. So they're quite distant. And down here you have the human common cold coronavirus. So MERS sits on its own. It's quite a different viruses from SARS and SARS-CoV-2. Now, what I haven't told you is that humans get the virus from camels, and a specific kind of camel, the dromedary camel. I'm going to talk to you about that in a moment. But at the moment, we see three clades of MERS coronavirus. There's clade A, B, and C. And clade B mainly is in the Arabian Peninsula, a few other places. It's the most diversified, and it's found in humans and in camels. Clade A, Saudi Arabia, Jordan, and UAE. Again, humans and camels. And clade C in other countries, Egypt, Morocco, Ethiopia, Burkina Faso, Nigeria, and Kenya. But most of the MERS originates in the Arabian Peninsula. There's no MERS in Nigeria or Kenya. The virus is in camels there, but there's no human MERS for reasons that we don't understand. The only human MERS is in the Arabian Peninsula. Everything else is exported from there, travel related. So the virus is spread from dromedary camels to humans. So we think that some years ago, maybe hundreds of years ago, the virus went from a bat to a camel, established itself in camels. I'm going to show you some evidence that camels have been infected for a long time. And then it infected people starting in 2012. Unfortunately, not all infections have a camel source. Many do, but not all of them. There's some spread from person to person in hospitals and within households. There's no community spread. There's none of this where The virus is shed for days before symptoms, and you can get it in mass gatherings and so forth. So we don't know why those cases have no camel source, but many of them do. Now, just to put this in the perspective of the Coronaviridae, right? The Coronaviridae family is divided into 4 genera: alpha, beta, gamma, and delta coronaviruses. And the viruses that we're talking about, MERS and SARS-CoV-1 and SARS-CoV-2, are all beta coronaviruses. And then you see some of the other species of coronaviruses are gamma and delta coronavirus, the human common cold coronas. Some of them are alpha coronas. Okay, so that's where these lie: MERS, SARS-CoV-1 and 2, beta coronaviruses. Oops, I went to a link. So as I said before, MERS, the disease, is only reported in the Arabian Peninsula and in travelers from this region. There's no MERS in Africa or Asia, even though they have infected dromedaries. Okay? There's no MERS disease. There have been 2 cases of MERS in the U.S. They've both been imported. However, in one study in Nigeria, camel abattoir workers were shown to be seropositive for MERS-CoV. So they weren't sick, but they had antibodies to the virus. So this is telling us that if you have close contact with camels, which you do in an abattoir for camels, right, you can be infected. But why it doesn't cause disease in Africa, we have no idea. I mean, it could be genetic basis, slightly different clade of virus. We just don't know. And it would be nice to find out, because understanding what controls infection and disease is important, right? So some of the signs and symptoms, this is a very typical severe acute respiratory disease. Most people have a severe respiratory disease, symptom onset 5 or 6 days after exposure, fever, cough, shortness of breath. Risk factors. There are a variety of diseases: diabetes, cardiovascular disease, chronic lung disease, heart disease, kidney disease, cancer, or recent travel in or near the Arabian Peninsula, or if you work in a healthcare setting in that area, especially if you had direct physical contact with camels, touching or grooming, or had close contact with a person who is sick with fever or respiratory symptoms. So these are all different risk factors for MERS. So basically, the CDC is not telling you not to go to the Arabian Peninsula. This is a very rare disease, but you should be aware of the risk factors. So here's an example of the incidence of MERS. So these are confirmed cases of MERS by place of infection and month of onset. This is from 2012, when we first discovered it, to July 2024. Been 2,622 cases, including 953 deaths. It's about a 35% case fatality rate. And you can see by the colors, most of these are in the Middle East. So here is 2012, when we first discover the virus, and there's quite a large outbreak in the Middle East, which eventually subsides mainly because of information and control measures. You know, we say camels are a risk factor. If you're going to work with them, you have to take precautions. But outside of the Middle East, you see mainly in 2015, big outbreak actually in South Korea. An infected traveler from the Middle East went to South Korea. He got sick. He was admitted to a hospital. They didn't know what he had. They didn't do any infection control. And so he infected a lot of other people in the hospital until they realized what it was. This is a epidemic curve of the cases in KSA from 2012 to 2023. And you can see the cases are in blue, peak in 2014, and slowly reducing as we got more aware of how to mitigate this exposure. And the deaths are quite high as well. But again, this is where the vast majority of cases occur. In fact, all of the index cases occur. Everything else is a travel-associated case. Nobody gets MERS in any of the other countries where camels have the virus. This is an older map, but I like it because it shows you the transmission in geographic range. Okay, so camels transmitted to each other throughout northern Africa, the Middle East, parts of India, Afghanistan, Pakistan. But the transmission from camels to humans and limited human-to-human transmission, largely in the Arabian Peninsula. And these red lines are exported cases to different countries. People who were visiting the Arabian Peninsula got infected and then went on elsewhere. There's no local acquisition of this virus, say in the U.S. or in different parts of Europe. So this is the dromedary camel. There are different kinds of camels and camelids, as you may know. I think let's move the. I can't move it. And here there's the Bactrian camel, domestic and wild, the dromedary. Then we have the guanaico, llamas, vicuñas, and alpacas, camelids. And these colored areas are the ranges of these animals. And the one we're concerned with is the dromedary camel. This is the only one that is endemically infected with MERS coronavirus. And you can see its range, northern Africa, Arabian Peninsula, and moving towards India. There's a little bit of, there are a few dromedary camels in Australia. They were brought there, actually. And there's actually a feral population of wild dromedary camels. But they're not infected with MERS coronavirus in Australia. It's a cool animal, right? It's just really neat. I like the way they look. Anyway, a number of studies have looked for seropositivity in camels. Either antibodies, some cases they looked for RNA by PCR, and samples were available back from 1981 through 2020. So you can see samples of camel serum. These are dromedary camel serum from 1980s are positive, are antibody positive against MERS coronavirus. So this virus has been circulating for many years, probably even longer than this, probably over 100 years in camels. And either it never infected humans or we didn't notice it. And then suddenly in 2012 it starts. But you see, antibody positive, these parts of Africa, interestingly, Kazakhstan, and then RNA positive, showing an ongoing infection most likely in Arabian Peninsula and parts of Africa. But again, the disease only occurs in the Arabian Peninsula, not in Africa, not in Kazakhstan. Very interesting. Now, here's a nice study, because you may be asking yourself, why does this happen all of a sudden? You know, the camels have been infected for hundreds of years with MERS coronavirus. Why in 2012 do we first see this? So a study was done in Qatar to try and understand the drivers of MERS emergence. Here's a reference for this here. It's done by Marion Koopmans and her colleagues. So here's the story. Basically, you know, Qatar became a wealthy country. Oil and gas exploitation led to an increase in the gross domestic product, growth in population over the 30 years, lifestyle transformed from Bedouin to urban sedentary life. You get increased obesity, you get other comorbidities, which are risk factors for MERS. And so then, in this wealthy environment, camel husbandry flourishes. People race camels, they have them as pets. And this exacerbates the desertification, that forced banning of free grazing. Camels are housed in compact barns alongside their workers, facilitating interaction with the people. And so this transition in husbandry leading to high-density camel farming, together with increased exposure to humans, increased camel movement for the racing and breeding industry, It's a convergence of factors driving spillover of MERS-CoV from camels to humans. It's our fault, folks. We did it. We moved camels around, decided they were cool to do things with, had more contact with camels. We got their virus. It's our fault. Here are two graphs that show this. Here's in Qatar, the increase in GDP over time from the 80s to the present. You can see it's going up. It's in US dollars. And the camel population is going up. And these are the total number of camels in this gray line. And then there's meat camels and milk camels. So obviously the vast majority are used for other purposes like pets and racing and so forth. So that's an interesting take on why suddenly you have this disease where the virus has been around for years. It doesn't tell us why it's not present in Africa, though. All right, so what can we do? So, there have been less than 3,000 cases total. Very hard to do anything for that. There are lots of experimental vaccines and antivirals. Chadox, the chimpadenovirus vector, has been used to carry the spike. mRNA vaccines encoding the spike of MERS-CoV, vaccinia virus vaccines, and many others. I mean, every kind of vaccine has been tested. None of them have been approved. It's not clear who would get it. There aren't that many cases. Maybe immunizing camels would be one way, a one world health approach, right? So the camels get infected at birth in their herd, and they don't get very sick. They get a little respiratory drizzle, very little lower tract disease, and it passes through the herd. But immunizing the camels has been proposed, but this is not a sterilizing vaccine. It's not going to prevent them from shedding. It's, in my opinion, not going to prevent transmission to humans. But how do you test that, right? So that's a tough one. So basically, no vaccine is approved. And there's some antivirals that we know, remdesivir, molnupiravir, they work against the virus in animal models. They haven't been approved. They haven't gone through clinical trials. I mean, the other problem is, how do you do a clinical trial? There are not that many cases, right? There are monoclonal antibodies that could be used therapeutically, but it's hard to test them because there aren't that many cases. So we're in a point where the only thing you can do is supportive therapy for MERS. So this summarizes now all the coronaviruses, where they came from. This is very cool. Here we have SARS-CoV-2, right? Which originated in a bat, probably some animals at the Huanan meat market, seafood market, civet cats and raccoon dogs, into humans. And in MERS, bats to camels to humans. SARS-1, bats to civet cats to humans. And then all the 4 common cold coronaviruses, some kind of animal, bats or rodents, through an intermediary to human. There's even a coronavirus of pigs, severe acute diarrhea syndrome coronavirus, which went from bats to pigs. Do you see the pattern here, folks? There's no need to say this came from a lab. In fact, there's no evidence. All right, so that's MERS. And if you want more virology, I have a whole virology course on YouTube. There's the kind of unwieldy link, but you'll find it there. Thanks for listening, folks.